Hello, and thank you for the opportunity to present to you at the meeting on the Mesa. My name is Tom Anderson. I'm CEO and Director of SwanBio Therapeutics, a gene therapy company focused on CNS disorders beyond the brain. We are located in Philadelphia, Pennsylvania, and Cambridge, Massachusetts, and have about 50 employees at our two sites. We are principally owned by Syncona Limited in London and Mass General Brigham Ventures in Boston. So how is SwanBio different from other CNS gene therapy companies? We have a singular focus on spinal cord related disorders, which has enabled us to become experts in biodistribution and in particular, intrathecal delivery. The commonality across our pipeline indications from a target tissue, symptomatology, and treating physician perspective means that we can adopt a modular approach to R&D, which is both highly efficient and cost-effective. Our lead asset, SBT-101, for adrenomyeloneuropathy, or AMN, is nearing the clinic. We fully expect to file the IND within the next 6 months and have the first patient dosed in 2022. We also recently initiated our prospective natural history study, the Cygnet study, which I will provide more details on later in the presentation. We decided to invest early in manufacturing because we wanted to ensure that it didn't become a bottleneck later in development. We also wanted to minimize the risk of needing to conduct a bridging study later in the clinical stage of the program. As a result, we have scaled a scaled commercial-ready manufacturing process and have successfully manufactured multiple GMP batches, including those needed to supply the phase 1/2 study. None of this would be possible without an experienced management team with a proven track record. Our leadership team has more than 90 years of gene therapy experience and has filed in excess of 100 INDs and 30 BLAs collectively. We have chosen to focus on the spinal cord as it is an uncrowded space with proven transduction, safety, and efficacy, as shown by the approval of Zolgensma for SMA. Our target diseases fall into 3 classifications. 1, spastic paraplegias, which includes our lead indication for AMN. 2, monogenic neuropathies, that includes indications such as Charcot-Marie-Tooth or CMT. And 3, polygenic neuropathies, including ALS, multiple sclerosis, and diabetic neuropathy. Our ultimate goal is to work progressively from monogenic towards polygenic indications with high unmet need and low competitive intensity in both the spinal cord and the peripheral nervous system by adopting a stepwise approach to risk. When we map our pipeline onto this chart, as shown by the green dots, you can see that we are well advanced on this path with 2 products that overexpress in the spine using proven technologies to replace a mutated gene with a normal copy. For dominantly inherited diseases, we are utilizing more complex suppress and replace approaches to knock down the mutant gene. In this particular program, we are employing 4 approaches, which not only increases our chance of success on this program, but also demonstrates our vast and varied technological capability in broadening our platforms. This has given us the confidence to initiate our first polygenic program, which while still early in discovery, is generating much excitement within our company and with our investors. When we overlay competitor programs against our portfolio, it is clear to see that the vast majority of programs are focused on the brain. And while there is clearly a lot of unmet need for these indications, the ability to safely deliver product through deep brain structures continues to be challenging. Our pipeline chart shows the relative status of each of our programs. A key point I want to highlight is that for our first 3 programs, which address monogenic indications, all have relatively large prevalences for rare diseases, with prevalence in the 8 to 15,000 range for the US and EU5, which translates to over 100,000 patients globally. This gives us confidence that we will be able to enroll patients in our clinical studies and also that there will be a significant market opportunity commercially. The indication for our polygenic program, SBT401, has a prevalence of 50,000, but unlike the above monogenic indications, has a much higher incidence with 20,000 new cases per year. In addition to driving our pipeline programs, we continue to seek ways to enhance and expand our technology platform. We strongly believe that it is important to be at the forefront of technological advances, and so we are always looking for ways to improve our biodistribution, lowering doses, and improve the safety profile through optimizing the expression cassette or by designing novel capsids and promoters. Our lead indication is for AMN, the most common phenotype of X-linked ALD, or adrenoleukodystrophy. It is a devastating neurodegenerative disease characterized by progressive muscle weakness. The disease typically leads to patients requiring a wheelchair within 10 years of symptoms appearing, and about 8,000 to 10,000 men are diagnosed in the US and EU5. The disease is caused by mutations in the single X-linked gene ABCD1, which codes for a peroxisomal transporter protein that shuttles very long-chain fatty acids, or VLCFA, into the peroxisome for degradation. In the absence of a functioning transporter, there is accumulation of VLCFA to toxic levels, which results in a dying back axonopathy, particularly in the long motor axons, leading to the loss of motor function. Onset usually occurs in men in their 20s or 30s, and in addition to the loss of mobility, it is frequently associated with incontinence and debilitating pain, severely impacting the quality of life for these young men. There are no approved treatments for AMN, and the standard of care is symptom control. Women, who were originally thought to be only carriers, have also shown to exhibit symptoms, although onset tends to be later by a couple of decades, and the phenotype is typically milder and slower to progress. Our preclinical program, in our preclinical program, we have successfully demonstrated targeted delivery, biological activity, and safety. In our initial in vitro work, conducted using mixed glial cell cultures from knockout mice, we were able to show a reduction of VLCFA to wild type levels. From there, we studied intrathecal delivery of SBT101 in a single knockout mouse model. We successfully showed dose-responsive ABCD1 expression and a significant reduction in VLCFA levels. This reduction increases over time, so we believe that the efficacy of SBT101 will be long-lasting. SBT101 also demonstrates a functional effect. In a double knockout mouse model that has a phenotype more representative of the human phenotype, we were able to show a significant improvement in grip strength with just 9 months of dosing. In our primate studies, we identified optimal conditions for IT administration and have demonstrated that we can achieve biodistribution and target expression levels throughout the spinal cord and DRG. Our pilot NHP tox study recently read out, and we were delighted with the safety profile achieved. There were no concerning tox findings, and the histopathology was indistinguishable from control at all dose levels, including the DRG and spinal cord histopath. Switching gears to clinical, we've made a significant investment to develop a deep understanding of AMN. We have strong connections with patient associations and KOL networks, which has given us tremendous insight into the burden of disease and endpoints that are truly meaningful to patients. To better understand the course of the disease, we have made a significant commitment to collecting natural history data, both retrospectively and prospectively. In the next slide, I will provide more details describing our prospective natural history study, the Cygnet study, that was launched earlier this summer. And of course, all aspects of our development path is guided by our interactions with regulators. To date, the 6-minute walk test is a commonly used endpoint in diseases impacting motor function While we believe that it is an approvable endpoint, we strongly believe that a more sensitive and clinically meaningful endpoint exists for AMN. The rate of deterioration in 6-minute walk test is about 10 to 12% over 2 years, and the rate of change is not linear. We believe that balance, objectively measured using a Romberg test, is a far more sensitive test for AMN patients, with changes of up to 60% seen in symptomatic patients within a year and 30% in asymptomatic patients. We believe it is also a more meaningful endpoint for patients since it is predictive of falls and the need for walking aids. We also plan to explore a number of biomarkers such as neurofilament light chain and electrophysiology, which we think will provide an early signal of efficacy. Our groundbreaking Cygnet study will be the largest natural history study conducted in men with AMN. We are targeting a minimum of 80 patients enrolled from across 8 to 10 sites in 4 countries, and patients will be followed prospectively for 2 years. We are running our natural history study with the same rigor as a phase 1/2 study. In addition to traditional office visits, we plan to make extensive use of digital data collection using wearable technology and telemedicine. By collecting tremendous amounts of data on each patient, we believe it will be easier to detect data trends, which could lead to earlier efficacy readouts in the phase 1/2. By comparing the measurement of endpoints using wearables at home and during office visits, we can validate this wearable technology ahead of their use in the phase 1/2 study. With this level of rigor, we believe that the Cygnet study will not only support our choice of endpoint to regulators, but may also be used to expand the phase 1/2 control group. With a 1 year head start on the phase 1/2 study, it should be possible to use the Cygnet study to pre-identify patients for enrollment into the later cohorts of the phase 1/2 study. We plan to initiate our phase 1/2 study in 2022. We've designed the study as a randomized placebo-controlled study, as we want to give ourselves the flexibility to convert this study to a pivotal trial. The primary objective of the study is safety, but we plan to explore the utility of novel endpoints tested in the Cygnet study. The study of 16 patients will be used to determine safety and the optimal dose for the pivotal trial. After patients in the low and high dose cohorts complete in 12 months, an interim efficacy analysis will be conducted. If the results from the interim efficacy analyses are compelling, we would seek to expand the maximum tolerated dose cohort and convert this study to a pivotal trial. Having readouts every 6 months after study start will provide lots of opportunities for agency interaction and also offers significant news flow for both patients and investors. We are currently engaged in fundraising a Series B and have already received sizable commitments from our current investors. The use of these funds will be to carry us through the first interim efficacy readout for SBT101 and a second IND for a pipeline product. We also plan to use the proceeds to further expand our pipeline and enhance our platform with novel capsids, targets, and promoters, thus creating additional value to our patent estate. We take this journey we are on at Swan very seriously, because for us, this is personal. I thank you for your kind attention to our presentation and wish you success during this conference.